When you only enter a gene for your input, you will see a total of 5 human genetics databases. You may notice that on the top right corner of each output box, there is an external link symbol. These symbols will take you to the original page where data are displayed. OMIM, or Online Mendelian Inheritance in Man, is the first database displayed. OMIM is a manually curated database that aggregates and summarizes information on genetic diseases and traits in humans. Human Gene Description box from OMIM provides a short summary of what is known about the gene. Gene-Phenotype Relationships provides any known disease or phenotype association with your gene of interest. This allows one to determine if this gene is a known disease-causing gene or not. Because there could be a time lag between the publication of a new disease discovery and the time that the paper is manually curated in OMIM, we advise that MARRVEL users perform a PubMed search to look into recent literature as well. Reported alleles from OMIM is a list of pathogenic variants curated by OMIM. EXACT is a large population genetic database of whole exome sequences of people who are selected to exclude severe pediatric diseases. EXACT provides overall statistics that we can use for gene prioritization. EXACT can be used as a control population database, especially for severe pediatric disorders, but requires caution. The control population gene summary box provides gene-level statistics such as probability of finding loss-of-function allele in the population. This is called the PLI score, and it may tell you how likely a single copy of loss-of-function of this gene may cause disease. The interpretation of a few numbers here are of interest when we try to prioritize which variants or genes of interest may be pathogenic or disease-causing. Looking at the PLI score of a gene has value, especially when we are dealing with dominant disorders, including those that are linked to de novo variants. If a gene has a PLI score of 0, it means that it is highly tolerant of loss-of-function variants. Thus, this gene is less likely to cause disease via a dominant haploinsufficiency mechanism. This does not, however, necessarily rule out other dominant gain-of-function related mechanisms. If a gene has a PLI score of 1, then it is possible that the loss of one copy is detrimental for human health. Geno2MP is a collection of samples from the University of Washington Center for Mendelian Genetics. It contains about 9,600 exomes of affected individuals and unaffected relatives with some phenotypic description. Here we provide a summary of what is found in Geno2MP in the top 2 boxes and also in the disease population box with a list of variants found in their database. If you suspect your gene of interest to be associated with your patient's disease, you may want to dig deeper and visit the Geno2MP website itself. We suggest that you click the link out button to the gene-specific page on Geno2MP. Here I'm clicking this link and it will bring you to a page that looks like this. Here we suggest that you filter for mutations that are similar to those of your patient and review the list of variants. We especially pay attention to variants with high CAD scores and click into the HPO profiles. For example, CAD scores that are higher than 20 are within the top 1% of all variants predicted to be deleterious, and scores that are higher than 10 are within the top 10%. HPO stands for Human Phenotype Ontology, which provides standardized description of human phenotypes. Here, check if the individual is a relative or an unaffected individual and see if the phenotypes are relevant to your patient or not. If these individuals are affected in the same organ system as your patient, you may want to contact the respective physician to see if it is an additional case that supports your hypothesis. Geno2MP provides an email form that you can use to attempt to contact the physician that submitted these cases. ClinVar is a database where researchers and clinicians submit variants with or without determination of pathogenicity. The top row provides a summary of the types of variants reported in ClinVar, and the reported alleles from ClinVar includes all reported variants and their review statuses. DGV is the largest public access collection of structural variants from more than 54,000 individuals. The database includes samples of reportedly healthy individuals at the time of assortment from up to 72 different studies. Variants listed in the DGV box are listed if they contain your gene of interest. This information may help you determine if your gene is duplicated or deleted in relatively healthy individuals. If you find that your gene is deleted in many alleles in the control population, it means that this gene is highly tolerant of loss of function variants. Thus, this gene is less likely to cause disease via dominant haploinsufficiency mechanism. This does not necessarily rule out other dominant gain of function related mechanisms. Possible limitations to these data include variation in source and method of the data acquired and lack of information regarding incomplete penetrance of pathogenic CMVs and whether individuals will develop associated diseases subsequent to data collection.